Good afternoon, good day, everyone. I'm very pleased to have you all here today for CanPath's March webinar. I'm Dr. Philip Awadallah. I'm the National Scientific Director of CanPath, the Canadian Partnership for Tomorrow's Health, and I'll be introducing today's speaker and also moderating today's session. So again, welcome and a thank you. So I'm very happy to be introducing Kimberly Skeed, who's a doctoral candidate in the Department of Molecular Genetics at the University of Toronto. And she's also an Ontario Institute for Cancer Research Rising Star, and where she's currently doing research and developing tools to identify patients at risk of cancer and heart disease. Kim is also the National Scientific Coordinator for the Canadian Partnership for Tomorrow's Health, where she supports the development of research and funding proposals, coordinates pan-Canadian scientific activities, and the publication of research reports, while identifying also opportunities for external research funding. So Kim is an extraordinarily busy person, and we're very happy to have her presenting today. So a little bit of bookkeeping or housekeeping today. So following today's presentation, we'll have a Q&A period. Please use the Q&A box. I'll be reading the questions out, and then Kim will be answering those questions. And so I encourage writing those questions during the presentation so you won't forget them and you'll have them ready for us. I'd like to do the land acknowledgement before we turn this over to Kim. So we acknowledge the traditional territories of the Mississauga of the New Credit First Nation, the Anishinaabe, Wendat, Huron, and Haudenosaunee Indigenous peoples on which the CanPath National Coordinating Center at the University of Toronto now stands. The territory was the subject of the Dish with One Spoon Wampum Belt Covenant, an agreement between the Iroquois Confederacy and Confederacy of the Ojibwe and allied nations to peaceably share and care for the resources around the Great Lakes. With CanPath teams working across Canada from coast to coast, we also acknowledge the ancestral territories that are home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. So it's with great pleasure that I turn this over to Kim. I'm very excited to hear her presentation. And Kim, you now have the floor.
Great, thanks so much. Can you hear me okay, Philip?
Yes, I can.
Perfect. Okay, thank you so much for the introduction. I'm really excited to have the opportunity to talk to you all today about some of the work that we've been doing to build capacity for Canadian cancer research using the Canadian Partnership for Tomorrow's Health project, or CanPath. Over the past few years, we've seen a shift towards more personalized approaches to improve health outcomes. Typically, what this looks like is we take populations of people and we profile them biologically. So we look at how unique differences in someone's DNA, their tissues, or even tumor samples can be used to stratify people into different treatment groups so that we can start to tailor therapeutic interventions to the individual in order to increase the likelihood of somebody having a positive health outcome. However, the idea of personalized approaches to understanding health outcomes extends beyond reacting to disease diagnoses with tailored therapeutic interventions, and also includes how can we better understand the unique factors driving the onset of disease in the population in the first place. And there are a number of key components that we can consider here, including environment, genetics, behavior, and lifestyle, age, and how all these factors work together to shape health risk over the course of our lives. Understanding the factors causing disease in the population is only becoming increasingly more important as rising levels of chronic disease are one of Canada's largest public health challenges. Current estimates project that 1 in 2 Canadians will die from cancer or chronic or cardiovascular disease, and 1 in 5 Canadians will be diagnosed with cancer, and up to 1 in 12 Canadians will be diagnosed with heart disease. Through focusing on uncovering the factors driving the onset of these conditions in the Canadian population, we'll be better equipped to prevent disease or identify it early and both improve outcomes as well as reduce the burden on the healthcare system. Longitudinal population cohorts, which observe the health outcomes of hundreds and thousands of people over time, are one of our best chances to understand the causes of health and disease in the population. Many countries have been investing in building large population cohorts or precision medicine initiatives over the past few decades. In Canada, CanPath is the largest population cohort and is able to work with other large cohorts around the world through the International 100,000 Cohort Consortium, IHCC, for example. Not only does CanPath advance health research in Canada, but it enables Canadian researchers to work internationally so that Canada can be competitive in health research. Today, I'm going to be chatting to you about how we at CanPath are building capacity to support Canadian research, with particular emphasis on some of the resources that we have been compiling to enable advances in cancer research. I'll start off by giving you an overview of the CanPath cohort, and then I'll walk you through how we've been leveraging our ability to do linkages to various data providers to identify a cohort of cancer patients in the national CanPath cohort. Finally, I'll end off with a little bit of a case study on how we can use these types of national resources to develop new approaches to identify cancer and cancer risk in the Canadian population in order to enable earlier diagnosis and interventions. To start with, the Canadian Partnership for Tomorrow's Health project is a longitudinal population health research platform which follows the health outcomes of 330,000 Canadians, that's 1 in every 100 Canadians, over time. The cohort was formed to assess the effects of genetics, behavior, health history, and environment on health and disease in the Canadian population. We collect a variety of different data types, including questionnaire data, physical measures, biologics, and a range of data is collected over time as well through longitudinal follow-up. At its outset, CanPath was mandated to follow the health outcomes of Canadians for a minimum of 50 years, and we've been doing so for the past decade and a half already. The cohort was originally established in 2008 with funding from the Canadian Partnership Against Cancer. In 2008, we also saw recruitment beginning. This is when we started our baseline participant recruitment. The first wave of data was made available to the scientific community in 2015. In 2018, we saw a change of our scientific home. CanPath was moved over to the University of Toronto and the Ontario Institute for Cancer Research. 2019 was an exciting year for us because we saw the onboarding of one of our newest cohorts and recruitment beginning in the Manitoba Tomorrow project. In 2020, we had a rebrand. We were formerly known as CPTP. We're now known as CanPath. Naturally, in 2020, with the emergence of the COVID-19 pandemic, we launched a number of COVID activities. The first wave of data, which was released in 2021, and we've had a number of subsequent data releases since then. And then as recently as a few weeks ago, we saw the launch of our newest cohort, Healthy Future Saskatchewan Research Project. The reason why I highlight the long history of the cohort is because it's a resource which continues to grow in value over time. After more than a decade of follow-up, CanPath has now entered an era of great informativeness and impact. To date, we've supported over 250 research projects and over 200 research publications. In the next 3 slides, I'm going to be giving you a little bit more of a granular overview of the data types that we collect. At the time of recruitment into the cohort, all of our participants complete a questionnaire which collects detailed information related to demographics, changes in health status, mental health, medical history, including prescribed medications and family history of disease. We also collect a number of different measures such as working status, household income, behavior, and lifestyle data. We don't ask participants to provide us with this information at the time of enrollment only, but we're also collecting this information routinely through the administration of detailed follow-up surveys, which really enable the collection and study of how these participant health outcomes are changing over time. Complementing our self-reported data collected through those questionnaires, we have a number of biosamples and physical measures which are also collected and available for a large number of CanPath participants, including blood, serum, as well as various macro measures such as blood pressure, height, BMI, and even more clinical data such as arterial stiffness. In addition to that, we have collected detailed environmental data from all participants, as we know that environment and our interactions with our environment play a major role in determining health outcomes. Beyond that self-reported data and those physical measures data alone, CanPath has also generated a wide array of omics data from our biosamples in order to support the genomic or genetic interrogation of health and disease in the cohort. Approximately 45,000 of our participants have been genotyped, and we anticipate this number to grow in the near future. But we also have a range of additional omics data sets, such as RNA sequencing, ATAC-seq, and methylome profiling that can layer on top of that, as well as imaging data for about 10,000 participants, inflammatory data for about 20,000 participants, and antibody profiles coming out of our COVID-19 work for about 28,000 Canadians. A strength of our cohort is that all participants are followed longitudinally, and this very long follow-up period is distinctive and essential to answer some of the most pressing questions around the factors driving changes in health and disease over the course of our lives. For example, we have a long history of supporting critical cancer research, really from coast to coast. So on the West Coast, you can see here we're highlighting some work led by Dr. Rachel Murphy, who has identified actionable behavioral changes that can be made to prevent lung cancer in never smokers. And these discoveries go right through to the East Coast, where we've seen work led by Dr. Trevor Dummer, who has highlighted the relationship between arsenic in drinking water and cancer outcomes in local populations. Beyond cancer and chronic disease research, we've also demonstrated that the cohort is incredibly valuable in the face of emerging health threats. Within weeks of COVID-19 being declared a pandemic, CanPath was out in the field collecting critical COVID-19 data, including things like exposure, risk, symptoms, and diagnosis. We also launched a national longitudinal serological surveillance program to study trends in immunity following vaccination and natural infection in the Canadian population. Our antibody study was enabled through the collection of longitudinal blood from around 30,000 individuals through using dried blood spot cards. And here we sampled from the general population, but we also targeted more high-risk populations, such as residents of long-term care homes, people living in areas which have a high prevalence of COVID-19, and people who are living in urban and rural communities. One of the key benefits of CanPath is that almost all of our participants have provided broad consent for linkages to various data entities, which enable the passive enrichment of the cohort with a range of different clinical, environmental, and other measures, all of which really facilitate our understanding of the drivers of health and disease in the population. For example, all of our CanPath participants have provided a postal code, which enables us to obtain a detailed snapshot into their unique environmental exposures. We've been able to achieve this through linking CanPath participants to the Canadian Urban Environmental Health Research Consortium, which captures a complex set of environmental factors for each participant, ranging from green space and walkability right through to levels of fine particulate matter in the air. Our linkages to environmental data have led to a number of key breakthroughs in our understanding of health and disease. In 2018, Dr. Philip Awodalla's research team reported that local environments, your daily exposures, are playing a larger role in determining patterns of gene expression and some cardiac outcomes than genetic ancestry does. So really saying that environment can trump genetic ancestry in terms of health risk in some contexts. Through linkages, we are also able to collect additional health outcomes from participants over time through linking out to hospital records or billing data, for example. Recently, we've seen a huge amount of progress in this area, thanks to a collaboration with the Canadian Institute for Health Information. We are currently linking the entire national CanPath dataset to the discharge abstract database in the National Ambulatory Care Reporting System at CIHI. Health outcomes data collected through CIHI will be hosted at CanPath alongside that self-reported health outcomes data collected through CanPath, and we're going to be able to make this data available to researchers at one central location. We're particularly excited about this collaboration as it will be the first time in Canadian history that this type of cohort data, biosamples, and national administrative health data will be available to researchers at this single location. And this represents a significant step forward in overcoming some of the historic barriers that we faced in Canada when it comes to bringing this type of data together. We anticipate that this CIHI data will be available to researchers in the fall of this year, so please do keep your eye out for that if you're interested in using some of that data. So this leads me quite nicely into the second thing that I want to spend some time on today, which is our work leveraging linkages to cancer registry data to build out a sub-cohort of cancer patients in the CanPath cohort, so as to better equip researchers in Canada to enable breakthroughs in cancer research. The construction of a national resource to support cancer research is naturally a major priority of ours, as cancer is the leading cause of death in Canada and poses an enormous health burden on both to the health of our Canadians, but also a burden on our healthcare systems. As I mentioned earlier, current estimates suggest that up to 2 in 5 Canadians are expected to develop cancer in their lifetime, which means that each and every one of us will be impacted in some way, either through being diagnosed ourselves or having a loved one diagnosed with cancer. We also know that our best chance at reducing this number, getting that number of Canadians who are experiencing these cancer diagnoses, either themselves or around them, is through prevention, so as to enable or early diagnosis and treatment. To do this, we need comprehensive data that can help researchers understand the factors increasing the risk of cancer and the development and enable the development of early detection biomarkers. Theoretically, Canada is really well suited to collate the data sets that we need to enable innovation in cancer research. And we see this. We know that Canada has an incredible wealth of cancer data, but one of the barriers that we continuously face is that all of this data is managed at a provincial level, and sharing and accessing that data can be incredibly cumbersome, costly, and there are a lot of barriers around where this data can reside. Historically, there hasn't been a single point of access to nationally harmonized health data that can be utilized by a range of different end users. But we think that CanPath is very well positioned to be a solution to this particular Canadian challenge that we're encountering, as we are already a cohort where we have nationally harmonized data housed at a single central location. We have collected over 1,600 variables related to disease outcomes across multiple time points, and over 350 of these variables are specifically related to cancer outcomes. And we've been capturing these outcomes longitudinally, both through our baseline questionnaire, but also through follow-up questionnaires and a number of COVID-19 questionnaires that we've launched in the recent years. Looking at our questionnaire data, we can already see that there are a large number of Canadians who report a history of cancer at the time of enrollment in the cohort. We're seeing that approximately 13% of our participants are reporting a history of cancer at the time of enrollment in the cohort, and we're seeing it emerging as one of the most frequently reported health histories in our cohort. The value of the self-reported data for cancer research continues to grow over time. We've supported over 77 publications, which have been specifically focused on cancer outcomes in Canada. And these have really generated a number of key insights into factors increasing cancer risk, including thinking about how various physical measures are able to impact the risk of early onset breast cancer. We've also seen studies which have focused on the impact of diet and lifestyle on shaping cancer risk in the general population. But we've also seen key breakthroughs in genetic research, so understanding how we can use select genes or combinations of genes to predict cancer risk in the Canadian population. But what we're recognizing at CanPath is that there is an opportunity to go beyond our self-reported cancer data and leverage our ability to perform linkages to really start to enrich the cancer data holdings within CanPath through partnering with cancer registries. And this is exactly what we're trying to accomplish with forming the Canadian Cancer Study. With linked clinical information, we can better identify which participants joined the cohort before developing disease and really understand their interactions and health trajectories with respect to cancer over the course of their lifetime. So through what do I mean by this? Through linking out to cancer registries, we're able to identify which of our participants in the cohort have never been diagnosed with a cancer, so that when at the time of their enrollment in the cohort, they didn't have that history of cancer, and they've been lucky enough to stay healthy and haven't been diagnosed with cancer to date. We can also identify individuals who joined the cohort with a history of cancer, meaning that they had cancer prior to joining the cohort, and providing us with that data and biosample. This is a really important group because it lets us understand cancer outcomes following treatment, but it also gives us a really good way to validate some of the self-reported health outcomes that we're seeing coming out of our questionnaires. But we can also identify this third group, which is comprised of participants who don't have a cancer at the time of enrollment and biosample collection, but who have subsequently been diagnosed with a cancer. From an early detection perspective, this third group is incredibly valuable, as we're able to obtain a snapshot into that individual's biology, sometimes years before cancer development, which gives us a really novel window of opportunity to understand the drivers of cancer in the population, and allows for the exploration of various risk markers that could have utility years in advance of current screening and diagnostic timelines. So we've been leveraging these provincial cancer linkages to start to map out the cancer outcomes in the CanPath cohort. All of our cancer data is collected and grouped according to the Canadian Cancer Statistic Reporting Guidelines. To date, we've been able to map cancer outcomes in the Atlantic provinces, Alberta, Ontario, and BC. As you can see here, we've been able to identify approximately 40,000 cancer diagnoses in the cohort. And we can also start to get a sense of not just how many cancers do we have fitting in each one of these various cancer groupings, but how these cancer cases are stratified across various demographic variables, such as biological sex, as well as age at the time of diagnosis. So what I'm showing you over here is the age at diagnosis indicated along the x-axis and the number of cases indicated along the y-axis. We can also start to stratify cases by incident and prevalence status at the time of enrollment in the cohort, as indicated by orange and blue respectively. And we can make this age association more granular and start to stratify age of diagnosis across those different cancer groupings so that we can start to see some of these age of onset or diagnostic trends emerging. Interestingly, because we have been around for so long right now, CanPath is now in a place where we have more incident cases than prevalent cases in the cohort. And this really demonstrates the value of following these participants over time and the power of these cohorts to really enable early detection and prevention research. So understanding which of our participants are incident and prevalent with respect to cancer status at the time of enrollment is critically important for us. Our linkages let us determine not only if a participant can be considered an incident or prevalent case at the time of enrollment, but also lets us ask, well, what is the time difference between enrollment in the cohort and cancer diagnosis? So what I'm showing you here is this broken down for two of our most common cancers, breast and prostate cancer. You can see the number of cancers are indicated along the x-axis, and the time between enrollment and diagnosis is indicated on the y-axis. The blue bars here represent the prevalent cases, so the time variable relates to the time between cancer diagnosis and enrollment in the cohort. The orange bars represent incident cases, so the time variable relates to the time between enrollment and cancer diagnosis. We can also start to ask what proportion of the cases that we're capturing have a biological sample available to support genetic research. The proportion of prevalent cancers with a sample available are indicated in the darker blue in our cohort, and the proportion of incident cancers with a biosample available are indicated in dark red. Here I'm just showing you breast and prostate cancer, as I mentioned earlier, but we can extend this right across all the cancer groupings that we're capturing. Related to biosamples collected prior to or following cancer diagnosis, we can also start to tease apart how advanced cancers were at the time of diagnosis. So what I'm showing you here are the proportion of biosamples for each cancer grouping broken down by stage at diagnosis. Incident cases are shown in red and prevalent cases are shown in blue. The stages are stratified by shade, ranging from the lightest shade, which indicates early stage of diagnosis, so stage 0 to 1, right through to late stage of diagnosis, stage 4, which is indicated by the darker shades of red and blue. One of the really important things that comes out of our ability to do these types of stage stratifications is really nicely illustrated with pancreatic cancer. Pancreatic cancer is typically only diagnosed at a very late stage, but what we're seeing here is that we're really getting a range in stages at diagnosis so that we can go back to the biosamples that those individuals provided us with prior to cancer development and start to interrogate them so as to better understand and develop novel risk and diagnostic biomarkers. So this really brings me to the last thing that I want to spend some time on today, which is a case study of how we can use cohorts like CanPath and the work that we're doing with the Canadian Cancer Study to identify some of the earliest initiating events in cancer evolution so as to better understand cancer risk based on individual molecular profiles. So to do this, we first have to understand the process whereby cancer arises in our cells. We can consider that biologically every single one of us is a population of cells. All of our cells are going to start off as healthy, and naturally as time goes by, we're going to start to pick up mutations. These are called somatic mutations. Most of these are going to be completely benign, meaning that they have no functional impact on the health of our cells. They're called passenger mutations and shown here in orange. But every once in a while, we're going to get a very rare event popping up called a driver mutation shown here in red that confers some sort of growth advantage to those cells, causing those cells to start expanding and forming that mass that we call a tumor. What we often do in cancer research is we can sample those tumors and look at the distribution of mutations in those samples to make inferences about how those tumors arose in the first place. What is the evolutionary history of those tumors? And oftentimes we turn to population genetics to understand how those populations of cells have changed over time. What Philip and I have been doing as part of my PhD research is taking some of these very classic approaches to understand how cancer arises, these population genetic approaches, and using them to understand how our blood changes as we age. The reason why we can do this is because the entire human blood hierarchy is rooted in a small population of stem cells which reside in our bone marrow. These cells are highly productive and are ultimately responsible for all the mature cells that we see in our blood. And naturally, because they're so highly productive, they need to be very tightly regulated in order to maintain the composition of the mature blood pool. But something which people have been paying a lot of attention to lately is this notion that as we age, we start to pick up mutations either in those stem cells or in early progenitor cells, which confer some sort of growth advantage to those stem cells, causing them and all of their derivatives to become disproportionately represented in the mature blood pool. Typically, when we see that this has happened to the extent that one clone is dominating our blood and is represented at a frequency of over 2%, we say that that individual has clonal hematopoiesis. And because this is a phenomenon which has been very closely linked with age, it's been termed age-related clonal hematopoiesis, or ARCH. This is something which is very common in the adult population, but has been found to associate with around a 10-fold increase in likelihood of later progressing to blood cancer. And it's thought that in those individuals who progress to cancer, they're accumulating these mutations in known cancer driver genes. And there have been a number of genes that we typically turn to understand risk of blood cancer, such as DNMT3A, TET2, ASXL1, et cetera. All of these tend to be recurrently mutated in this particular blood phenotype and are associated with later disease progression. These mutations take a range of different forms, ranging from single base pair changes called point mutations to disruptions at a chromosomal level, where we're seeing large structural changes in our chromosomes. But the real conundrum comes in here, which is if we look at the proportion of individuals in the population who are healthy and harbor one of these mutations in a known cancer driver gene, which I'm showing you here in gray, compared to the proportion of individuals who eventually progress to disease, shown in red, those numbers look really different. And there's some fairly massive implications to this at a population level. For example, if we start responding to all of those individuals shown in gray, we're going to end up hugely overtreating a proportion of the population that never really requires a therapeutic intervention in the first place. But if we don't respond to those individuals falling into those red bars, those individuals who do eventually progress to disease, we're missing out on an incredibly valuable opportunity to capture diseases in their infancy when treatment outcomes are more likely to be effective. What we are proposing is that the genetic background in which these driver mutations matters, that basically it's not just those driver mutations, it's all the other mutations that are harbored in our blood cells. By that, I mean, we have a very good idea of what positive selection looks like when it's acting to increase the frequency of that single driver mutation in a population, causing that clonal expansion, which often leads to tumor development. But we also have a really good idea of what negative selection looks like when it's removing mildly damaging mutations from our cells in order to maintain the integrity of our tissues. This is a key foundational biological process that happens in our cells all the time. But what we don't quite understand is what happens when we get seeing those positive clonal expansions that are associated with cancer, when we're seeing those positive signatures acting on cells that have that competing negative selection acting on passenger mutations. We might start to see competing evolutionary pressures emerging between all of the mutations harbored in our cells that could be inhibiting later progression to disease. Of course, we also can't discount models where our cells are exhibiting these clonal expansions, or undergoing these clonal expansions purely by chance. This is something which is called neutral evolution or genetic drift. And most of the time when we're looking to understand the earliest stages of cancer evolution, we really focus in on these models of neutral drift, where things are, any changes in our cells are governed by chance, or positive selection only. So our team has previously shown that there are a number of different interacting evolutionary pressures which shape blood health as we age. This came out in a paper in Nature Communications about a year ago. Here we built a machine learning classifier to evaluate the distribution of mutations in a blood sample. and use that distribution of mutations to predict those selective pressures in blood. We applied our machine learning classifier to blood sampled from healthy individuals, as well as blood sampled from individuals about 7 years before they progressed to blood cancer. And what we found is that immediately the vast majority of individuals are really rejecting these simplistic models of positive only or neutral evolution that we typically use to understand the evolutionary trajectory of cancers. And we're finding that many individuals are exhibiting these signatures of negative selection. And interestingly, this negative selection is playing a major role in preventing progression to acute myeloid leukemia. So you can see here that individuals who fit those neutral or negative models of evolution are remaining AML-free for much longer, which is really highlighting the utility that some of these modeling approaches might have for understanding cancer risk in the general population. So right around the time that we're making this discovery that cancer risk is associated with different evolutionary models in our blood, a study came out of the UK Biobank, which is a UK population cohort, which looked beyond the small single base pair mutation changes that we've been often exploring in the context of clonal hematopoiesis, and rather looked at larger structural changes which were accumulating in blood of largely quite healthy individuals. So typically these structural changes tend to fall into 3 categories. They're termed mosaic chromosomal alterations, and they can take a number of different forms. So instances where we are losing DNA, so we're losing large segments of our chromosomes, instances where we're gaining DNA, so places where we're gaining large segments of chromosomes, or instances where we're losing heterozygosity, so we're losing variability in our genomes. But the key statistic that jumped out at us was that these large chromosomal alterations were present in the blood of approximately 5% of the healthy UK population, which raises a really interesting question from a selection perspective, which is, if selection is playing such a major role in maintaining the spectrum of mutations we see in our blood, why are some of these large mutations tolerated to such a great extent at all? So to investigate if this observation held true in different population contexts, we turned to the CanPath cohort. It's an incredibly valuable resource with which we can use to ask some of these questions, as we have those biosamples, we have genomics, but we also have those health outcomes data available, which lets us not only map the genetic events, but also query their impact on our later health outcomes. So through CanPath, we were able to access genotyping array data for about 14,000 individuals to call these somatic events in the Canadian population. So what I'm showing you over here is a plot where you can see the chromosomes indicated along an outer ring and the locations of these structural changes. So those losses, gains, and CNLOHs indicated by the bands of blue, green, and orange respectively. One of the benefits of CanPath is that we have access to a variety of different genotyping array technologies, which lets us start to tease apart some of the impacts of those technologies that we're using on our ability to make these biologic inferences. For example, you can see here right away, we're capturing a much, much higher prevalence of these somatic MCA events in the general population. We're seeing that up to 14% of individuals are harboring one of these large structural changes in their blood. And we think that this difference in prevalence is largely due to the fact that we're using a much denser genotyping array. And with that, we have more resolution to capture smaller structural changes in our blood systems. And as you can see here, where I'm showing you the density of each array along the bottom and the distribution of MCA lengths along the side here, we are finding that we're able to identify those small MCAs that would have been missed with our denser genotyping arrays. That said, the length of these events becomes really interesting from a selection perspective as well. Presumably, large MCAs are going to impact more of our genes, more of our genetic material, and be more damaging and have a larger fitness impact, both on our cells, but also on our health. And this is something that we can test quite directly within an individual through comparing the length of these MCAs with something called cell fraction, which is asking what proportion of our cells are harboring these large structural changes. In the absence of selection, if these changes are truly neutral, we might expect to see something like this, where there is no real association between cell fraction and the length of these structural changes in our blood systems. But what we're capturing in our CanPath data is this really nice relationship where we're finding that the size of these MCAs, these chromosomal events, are negatively correlated with their frequency in our blood. And what this is telling us is that there is a functional consequence to these mutations, and negative selection is playing a role in limiting the expansion of large clones harboring these large variants. And it's only those really small MCA events which are able to rise in frequency in our blood. Of course, the size of these MCAs is not the only consideration with respect to the impact of these events on our health. And we also need to think about which areas of the genome these MCAs are impacting. And to address this, we implemented a genome-wide hotspotting approach where we can scan the genome looking for regions of where we find a disproportionately high burden of structural variation. So what you can see here is each box represents a chromosome. Genomic locations are indicated along the bottom, and regions which are significantly enriched for these MCA events are indicated along the y-axis as peaks. For example, we can zoom in on chromosome 13 over here, where you can see that we're capturing this really nice enrichment of loss events on the Q arm of chromosome 13. And interestingly, this peak is actually popping up over DLU2, which is a region which has previously been implicated in ARCH and pre-cancer phenotypes, as it plays a role in the development of chronic lymphocytic leukemia. We're actually finding that broadly, these MCA peaks are often accumulating over genes which are either ARCH or cancer associated. For example, here on chromosome 2, you can see that these peaks are hitting genes like AXXL2 and DNMT3 that I mentioned earlier. And naturally, this accumulation of MCA events at genes which are implicated in cancer raises a really interesting question about whether or not these individuals are in fact at risk of progressing to blood cancer. The first question that we can ask to start to interrogate this is, well, are the individuals who harbor an MCA that impacts one of these cancer-associated genes, are we seeing an increased clonal frequency? Are we seeing more successful clonal expansions in these individuals, which would be a way that we could interpret cancer risk? And yes, what we're finding is that in individuals where they have an MCA hitting one of those hotspots of those cancer-associated genes, we're seeing that those individuals tend to exhibit significantly greater clonal expansions, telling us that these individuals might be at an increased risk of cancer development. But we can also validate this further through turning to resources such as the Canadian Cancer Study, which rely on cancer linkages to determine the downstream impact of those mutations on cancer outcomes. So specifically, what we were interested in asking here is, are those individuals what we're interested in asking here was, are those individuals who provided us with a blood sample before progressing to cancer, those incident cases, are those individuals at an increased risk of progressing to cancer if they have one of those MCA events? So that's this top group that you can see over here, where those individuals provided a blood sample years prior to progressing to blood cancer. And when we look at patterns of MCA accumulation in this group, we're finding that individuals who harbor an MCA are at a significantly greater risk of progressing to blood cancer. For those of you who are new to survival analyses, what I'm showing you here is the time until cancer development on the x-axis, and the proportion of individuals in each group, I group by the number of MCAs per individual, who remain cancer-free, shown on the y-axis. As individuals within each group develop cancer, those lines will start to drop. And you can see here that a greater number of MCAs per individual is corresponding to a greater drop or more individuals progressing to cancer. And you can see how this risk really scales with the number of MCAs per individual. This hazard ratio is increasing in individuals who have 1 to 2 MCAs compared to individuals who have over 3+. We see that risk of progressing to cancer shooting up to 5 and 26 respectively. I'm going to stop there. We are further trying to understand how some of these molecular changes can be linked to eventual cancer diagnosis through interrogating some of their impact functionally and understanding their downstream impact on gene expression and protein production. But what I really want you to take away from these stories is that how these types of large population cohorts really build capacity for cancer research in Canada. As time goes by, these types of activities, which recruit and follow the general population, will only continue to become increasingly more valuable and can really be leveraged as a foundation for more niche nested cohorts within the larger population cohort. Today, we've demonstrated some of the advantages that CanPath holds for building out cancer research capacity in Canada. But CanPath is also focused on health and disease more broadly. So we hope to be able to extend many of the approaches that we've spoken about today to other conditions as well. Finally, we're building this resource. We want you to use it. So you are able to go and submit an access request to get access to data and biosamples at our access portal. So I put the site up there, so portal.canpath.ca. Finally, I'm going to end off with saying that CanPath is really enabled through a number of incredible teams, incredible researchers, and is fundamentally a partnership of health institutes from coast to coast. So thank you to everyone who has been involved, and most importantly, thank you to all of the Canadians who very generously donate their time, information, and biological samples to building these types of platforms for research. So with that, I'll take any questions. Philip, I think you're going to read some of them out, right?
Great. Thank you very much, Kim. That was a wonderful presentation. And I'll just start with a comment to just say it's been wonderful working with Kim, both as her PhD advisor and working with her on the CanPath activity. We couldn't have gotten much of what we've done with CanPath over the past 6 or 7 years without Kim's unfailing support. So this was a great presentation, Kim. So we have 3 questions that I'm going to read out. And please, I'll encourage the audience, if you have questions, type them into the Q&A and I'll read them out and Kim will answer them. I think this first question, Kim, really speaks to the question of infectious disease and cancer, or what can we do to support infectious disease as a potential causal relationship with cancer. So Zara Mazruri is asking, how have you come across significant relationships between, say, Helicobacter pylori and gastric cancers? So Kim, over to you.
That's a great question. I think, so this is not my area, so I specifically work in blood cancer, not in gastric cancer at all, but I do think that there have been reports of an association between H. pylori and gastric cancer onset previously. So I guess my answer to that question is I would encourage you to check out our data and see if you can interrogate that association yourself. We do collect a broad range of data related to infectious disease exposures, prior health histories, that could be used to start to tease apart that association. And I think that that would make for a tremendously interesting study.
Great. Thank you very much, Kim. I'm going to start with a question. In that picture you showed of the cancer incidences with respect to prevalent and incident rates, the prevalence were at the tops, the incidents were at the bottom, and then there were some people at dots. Both. I presume that extended for both. I think you're only showing one cancer, mostly a single cancer event, but presumably there's other data with regard to multiple diagnoses being captured as well.
Yeah, so I think the figure you're talking about, Philip, is where we're mapping the time between diagnosis and enrollment in the cohort.
I think that's a really compelling figure. Go ahead.
Yeah, and absolutely. I think we are able to capture every diagnosis that an individual has, and that's the benefit of these types of linkages in enabling cancer research, is that often the entire cancer journey is going to be shaping some of the biological outputs that we're collecting in CanPath. So it's important for us to understand, if we're sampling blood from an individual, have they had a cancer treatment? How might that be impacting some of the blood profiles that we're capturing, or even just their genetic landscape rather. So yes, we're able to capture an individual's entire trajectory of interactions with the healthcare system with respect to cancer, to really support not only questions around, what is the risk associated with particular genetic markers, but how might things like treatment be shaping the prevalence of those genetic markers and risk in the long term.
Fantastic. I'll move on to another question from the audience. Roland Hussein has asked, do the provinces use a standardized system for doing objective tests and subjective data from questionnaires to help in linkages? Are validation studies conducted?
Yeah, that's a great question. So I think I touched on this when I was mentioning going through the 3 different groups. So saying that people could have, we could have instances where people were healthy and have been lucky enough to not been diagnosed with a cancer throughout their entire participation in the CanPath cohort. But we also have instances where people are coming in and reporting that history of cancer. And that group is really valuable because it lets us actually start to validate some of the results that we have coming in through our questionnaire data. So In 2015, there was some work done by Craig Earle, who's now at CPAC, who is actually looking at how using some of these linkages to effectively validate the rates or the reporting of cancer through the self-reported health outcomes data. And largely those reports were tracking really well with what we were capturing through clinical linkages. I think there were some instances where we saw some over-reporting of cancers, but that was largely in the area of skin cancer.
Wonderful. Shiraz Kima is asking a very important question. Will linkage to CAIHI's administrative data allow researchers to apply more AI and ML techniques, which often requires large sample sizes?
Yes, absolutely. I think particularly for machine learning runs in a space where more data typically leads to the discovery of more outcomes. Definitely, CAIHI is enriching the number of clinical outcomes that we're able to observe in our cohort. That being said, often to access CanPath data, we do ask that there be a hypothesis. So there be some level of hypothesis-driven research rather than data mining. But we're always open to conversations about how we can be applying some of the most innovative technologies to lead to health breakthroughs.
Excellent. I'm going to, seeing as you've done so well with timing, we have the questions are pouring in fact, but I'm going to throw in another question in here. Could you maybe illuminate a little bit more on the kinds of data assets that we are seeing or capturing with regard to, say, not just diagnoses or say ICD scores, but other information that we might be capturing as well? With respect to cancers, for example, yeah.
Oh, right. So I think what Philip is getting at is that we are largely taking a three-pronged approach to map cancer outcomes in the cohort. So a lot of the first step of which was what I was talking to you about today, which is our efforts to collect these aggregate reports from our regions to start mapping out what do we have in the cohort. But we're also able to collect some cancer data through our collaboration with CAIHI, for example. So there we're largely getting hospital billing information. So yes, we do collect some cancer data, but the real wealth of data for cancer is going to come from linking to cancer registries. So CanPath broadly, the provinces, we are able to do linkages between the provincial CanPath cohorts and cancer registries. We're tackling this at a national level now. Through those linkages, we're able to obtain a wealth of information ranging from ICD code level diagnoses through to treatments, pathology records in some cases either. In many locations, we do have consent to even be able to link out to pathology biobanks so that we're able to obtain samples that were taken at the time of diagnosis from participants. So there's a real wealth of data that's out there, and hopefully this can all, create a better snapshot of a participant's biological profile and help us understand cancer risk.
Noor Breck has asked, thank you, Kim, for has commented and asked, thank you, Kim, for this super informative and outstanding presentation. Related to mapping cancer cases in CanPath with a linkage perspective, I'm wondering if you consider drug data linkage in building the Canadian Cancer Study, knowing that drugs are one of the most used interventions by healthcare providers. Increased drug utilization patterns in Canada thus could be a critical predictor, risk factor, and significant covariate to consider in cancer studies, including pharmacoepidemiological cancer studies. Thank you again for a great presentation.
Yeah, that's a really important question, and absolutely. So there's been a lot of work done, particularly with our self-reported drug information. So we do ask participants to report that as free text. Naturally, that introduces another whole challenge of processing that data. And Sasha Bernatsky has done a lot of incredible work in that space. At CanPath, we're actively thinking about how we can be collecting that data through linkages, and we have a number of ongoing conversations. So I think you're really hitting the nail on the head with respect to some of our priorities right now. So thank you for that question.
Great, Kim. We're moving over to friends and family at this point. Donna Turner has asked, are there plans to use some of the other lifestyle data collected by CanPath to understand how this impacts what you see in the genetic cancer risk analysis? I think specifically referring to the latter third of your presentation today. And how would we do this? So.
Yeah, absolutely. That's a really important question. I think particularly in the blood cancer space, there's been a lot of interesting work looking at how various environmental exposures might be impacting these ARCH phenotypes or cancer risk. So there was recently a paper which came out in Nature from Regeneron Genetics Center that was looking at the association between these arch phenotypes and smoking. For example, Peter Van Galen has also done incredible work looking at how sun exposure might be actually impacting some of these patterns of somatic mutations we're seeing in our blood. So absolutely, there are, the more data that we have, the more holistic of a picture we can get about why some people are progressing to disease and why other people are remaining healthy. And that's certainly the question which is motivating why we went down this road of understanding the evolutionary histories, because I think in cancer research, we often tend to adopt a little bit more of a driver-centric lens, understanding how those cancer driver mutations are leading to outcomes, but recognizing that, we are a constellation of somatic mutations and cells, and we need to think about how all of those mutations impact health outcomes, as well as the factors driving the accumulation of those mutations. The last thing that I'll say, just to answer Dana's question, is I think we're, all reading some of the recent reports that have come out about the impact of alcohol in shaping cancer incidence. So that's something that we're tackling quite directly in Philip's lab right now.
I'm going to ask you to put on your international hat for a moment and think a little bit about the Canadian Cancer Study in the context of, strengths and, say, challenges compared to, say, potentially other jurisdictions like in the United States or in the UK, both from the perspective of capturing the data linkages, but also potentially from, what can we do with longitudinal collections of potentially blood samples and so on. So, care to comment?
That's a really important question. I think that this is what I was getting at when I was talking about the Canadian challenge, right, with respect to how we bring together data. Canada does have such a wealth of data resources compared to many, many other countries, just by virtue of how our health system is set up. Our biggest barrier to really being able to benefit from bringing together those resources is really our inability to share those data across provinces. So that's where initiatives like CanPath, which are seeking to bring together provincial data into one central national location, do become incredibly important. That's why we're thinking about how we can leverage provincial linkages to really build out that national data asset as well, because as soon as we do that, we get into a different space with respect to power, with respect to the types of analyses that we're able to support, because we just have more data, so we can identify some of those rarer associations as well. And I think that holds true for international collaboration as well, right? Cancer isn't something which tracks along borders. The more that Canada is able to collaborate with other international precision medicine programs, the more progress that we can make in that arena as well.
Fantastic. Zvi Aviv has asked two questions. I think we've answered the second one. Are you tracking over-the-counter medication used by participants? And same for prescription medications. That's the second question. So.
Yeah, that's a great question, and we do ask participants to self-report any medications that they're taking, so we are able to track it in that way. And as I mentioned earlier, we are thinking about how we can move beyond that self-reported data in that space alone, and actually start leveraging linkages to enrich that particular aspect of our data.
So we are at the end of questions, Kim, and I'd like to thank you once again for a wonderful presentation. The research program that you've described, I think, is going to be very exciting, both for Canadian scientists and international scientists, and I think it's an exciting development for CanPath, specifically the Canadian Cancer Study, and of course your research is significant, both in understanding how blood cancers evolve and demonstrating how we can use a cohort like CanPath to facilitate that type of science, which I think is a paradigm shift in the way population cohorts are now being used for science. So, Kim, I'm going to thank you once again. And if you have any questions, please contact us at CanPath. Megan has included information about our mailing lists, how you can reach out to CanPath, how you can follow us on social media. And if you have any more questions for Kim, I'm sure she'd be very happy to answer them. And again, Kim, thanks very much for a great presentation.
Thank you.